Luca, our last universal common ancestor, dwelled during a time when the atmospheric composition was primarily carbon dioxide, methane, nitrogen, and most importantly hydrogen gas, which came from volcanoes and metamorphism, and it gives a red tint to the sky. The temperature is very hot, it's 49 to 88 degrees Celsius. We are at the ocean surface and not hydrothermal vents. The environment has organic compounds and molecules such as sugars that Luca will use as energy. So let's take a closer look and dive deep into who Luca was. Luca has peptidoglycan surrounding the cell membrane, and it's gram-negative as we can see here because it was stained a color that is pink or red. There are actin-like protein filaments making up Luca's cytoskeleton. There is a single circular chromosome with roughly 2.75 million base pairs, and there are ribosomes within the cytosol or cytoplasm available to conduct translation throughout the cell. Luca's metabolic pathways such as glycolysis and carbon fixation, or more specifically the Wood-Youngdahl pathway, along with a modified TCA cycle, rely on a network of enzymes and complexes that facilitate the energy transfer. Not to mention there is an ATP synthase and a minimal form of the electron transport chain. Hydrogen gas diffuses through the cell and into nickel iron hydrogenase, where the gas is split into 2 protons and 2 electrons. Additionally, 2 molecules of carbon dioxide also diffuse through the cell membrane. One goes through a series of enzymes and complexes and is reduced with the help of electrons from the hydrogen gas that was split. The other is converted to carbon monoxide by carbon monoxide dehydrogenase. They meet together with coenzyme A to enzymatically form acetyl-CoA via acetyl-CoA synthase. Meanwhile, glycolysis is also going on and forming acetyl-CoA. Acetyl-CoA enters the 3-enzyme modified TCA cycle. It meets with a 4-carbon molecule at homocitrate synthase. Out comes a 6-carbon molecule that enters succinate dehydrogenase, which spits out a 4-carbon molecule that enters 2 subunits of ferredoxin oxidoreductase. This all leads to the advent of ATP and the generation of a proton gradient that powers our limited ATP synthase that has only beta subunits instead of beta and alpha subunits connected to the gamma subunit.